Let's begin. Hello, and welcome to today's webinar. I'm Lisa, and I'll be your moderator for today's session. Before we get started, I just want to give a few quick housekeeping notes. Today's session is being recorded. We will send the recording to all registered attendees, so don't worry if you miss a portion or if you want to review it later. You can submit questions for our speaker at any time using the Q&A box that you see on your screen, and then we'll have a live Q&A session with Dr. Stern at the end of the presentation. We'll have a short 3-question survey when you leave the webinar, and we would greatly appreciate receiving your input. Let's go ahead and get started. Today's topic is a timely one, shaping how we study disease, evaluate therapeutics, and reduce the reliance on animal testing. Whether you are working in drug discovery, toxicology, or regenerative medicine, understanding how to build and optimize 3D cellular systems is increasingly vital. It's my pleasure to introduce our expert speaker on this very topic, Dr. Alyssa Stern. Dr. Stern is a scientist on the applications team at Cell Microsystems. She holds a PhD and a master's in physiology from NC State, as well as a bachelor's degree in animal science. Her background in comparative physiology and cell biology has focused on developing both 2D and 3D in vitro models for drug and toxicological studies. At Cell Microsystems, she's leading the development of new cellular workflows, especially in 3D model systems like organoids. She'll be sharing her insights today on organoid workflows, common challenges, and how to streamline your 3D model development for more reproducible and biologically relevant results. With that, let's turn it over to Dr. Stern. Alyssa, the floor is yours.
Thank you, Lisa, for that kind introduction. And thanks to everyone joining us online today. We really appreciate your time. And I'm very excited to talk with you today. In today's webinar, I'm going to show you how scientists are leveraging a proven technology called the Zero3D that simplifies and standardizes the development of tissue-specific 3D models. To do this, I'll be reviewing some case studies on how these scientists are developing novel organoids to drive their discovery, and also discussing why 3D cell-based models offer more predictive power for drug discovery. This is the canonical pathway to drug approval. Each step of this drug development process has been rigorously optimized and validated for decades, yet somehow the overall success rate is still less than 10% for a drug to be FDA approved. In the traditional path, 2D immortalized cell lines have been used for early discovery, and animal models have been used for preclinical development. And a large portion of time and resources are spent in this translation gap, sometimes referred to as the valley of death, where animal models are employed to make drug efficacy and safety decisions. The use of animal models really brings about 2 key problems. The first being, of course, ethical concerns behind animal use. But the second is that animal models do not recapitulate methods of exposure, modes of action, or metabolism of humans. And it's been proven over and over again that drug efficacy and safety in animals does not align with clinical outcomes. One example of this, there are medications that are generally recognized as safe, like aspirin, that may have never passed animal testing. And on the other hand, as stated here with this low success rate, many drugs fail to make it through these preclinical development processes and are ultimately withdrawn because of safety or mortality concerns. Now, this is largely due to fundamental physiologic differences between humans and animals. And this leads to a very urgent need for more relevant model systems for drug discovery and safety testing that allow us to test our drugs more rigorously and more efficiently than using animal models. So it was 1959 when skepticism of animal use first came about, and there was legislation to reform the drug development process. This first initiative, referred to as animal use alternatives, or commonly known as the 3 Rs, set out to reduce, refine, and replace animal testing in the drug discovery process. For refinement, when animals had to be used, it was recommended that husbandry and experimental procedures utilizing animals were modified to minimize the pain and distress. In addition to that, there were outlines for experimental design modifications, as well as ensuring researchers were using proper statistical evaluation in order to allow them to use as few animals as possible. And then finally, for replacement, shortly after this initiative is when the first cytotoxicity assays were being developed using immortalized cell lines to replace the use of animals. In addition to all the ethical concerns, we have decades of data that shows there are major safety concerns involving the use of animals for drug testing. Recently here in the US in 2022, the FDA Modernization Act 2.0 was the first major legislation to be passed that authorized the use of non-animal models, including cell-based assays, computer models for new drug applications. This act also removed the requirement to use animals for testing biosimilar drugs. At the beginning of this year, just in the last few months, a proposal has been made for the FDA Modernization Act 3.0, which builds on 2.0 by establishing funding programs to implement and validate new model systems to accelerate the incorporation of these new approach methodologies. And this proposal is why we really felt the time is critical to start discussing these new approach methodologies. These can be in silico tools and computational modeling, ex vivo tissues, and then what we'll be discussing today are the in vitro human-derived systems, which can not only be used for testing, but can be paired with some of those other methods like computational modeling to more accurately model clinical dosimetry and mechanisms of action. So as I briefly mentioned, one of the main issues with the historical use of 2D immortalized cell lines is that these cell lines no longer accurately represent humans. There's genetic drift in these cell lines. They oftentimes have non-physiologic behavior. And because of these things, many times they either over or underestimate drug sensitivity. So the lab-based human models really start with the broader acceptance of induced pluripotent stem cells as an alternative to traditionally used immortalized cell lines. iPSCs are adult cells, either derived typically from skin or blood, that can be genetically reprogrammed back into a pluripotent state, which means they can then develop into nearly any cell type of the human body. One advantage of iPSCs is that, of course, there are fewer ethical concerns with them compared to previously used embryonic stem cells, but also they can model very rare and complex human diseases. In addition to this, they can be established from a variety of patients that represent the diversity of our population and therefore more accurately represent the clinical setting. In the last decade or so, there's been a large push to develop methods and protocols to differentiate these iPSCs into functionally relevant cell types of virtually any tissue in the body. And now the FDA is promoting the use of these lab-based models that can mimic human organs, such as liver, heart, or neuronal models, which are important, of course, for our efficacy and safety testing, as well as deriving 3D models from tumors to more accurately test the drugs that are being developed. Within the context of using these iPSC-based models is this continuum of in vitro systems that can be used throughout the drug discovery pipeline. So the most simplistic, of course, being 2D assays. This is where iPSCs would be differentiated into a cell type and can be used in a high throughput method for early development, including hit screening and lead optimization. So while these iPSC cells are are great for these high throughput screenings, it's well known that 2D cultures don't have the multicellular complexity or the 3D organization of cells that mimic tissues. On the other end of the spectrum are organ-on-a-chip technologies. These are known to better simulate the structure and function of whole tissues. Sometimes these organ-on-a-chip methods mimic fluid flow or mechanical forces, and many times researchers are able to combine organ-on-a-chips with various tissues throughout the body to create this idea of a body on a chip. Now, of course, these organ-on-a-chip models offer much greater complexity and physiological relevance, but they're a lot harder to generate, and they're generally considered to be low throughput, and therefore are better employed at the later phases of preclinical development. So for the purposes of today's webinar, I'm going to be focusing on 3D model systems that offer the greatest potential for utility to be used throughout the development process because they marry the benefits of throughput and physiological relevance. In the context of these 3D culture models, there's really two main classes, spheroids and organoids. Spheroids can be formed from a broad range of predetermined cell types. They are scaffold-free aggregates of one and sometimes multiple cell types if you're doing co-culture. They may or may not exhibit polarity based on the cell types that are there. And really the main benefit of spheroids is that they can be very easily established, they're scalable, and oftentimes the methods are amenable to automation. Organoids, on the other hand, are derived from stem cells. These are either the iPSCs we've discussed, adult stem cells, organ progenitor cells, and these cells, through cell signaling cues, can be directed down tissue-specific lineages to contain multiple cell types, just like our tissues do. These are self-organizing, and sometimes they require scaffolds like extracellular matrices or hydrogels. They exhibit polarity, cell migration, and have a tissue-like organization. And ultimately, this makes them much more in vivo-like in terms of their cellular complexity and their architecture. And in addition to that, they can be derived from specific patients and used in applications such as personalized medicine. To culture 3D models, there are two main classes, static and dynamic culture systems. Probably the most commonly used and well-known is dome culture or some other form of embedding cells in an extracellular matrix or hydrogel. Organoids and spheroids can be formed in ultra-low attachment environments. These are specialized plates that have an anti-adherence coating, and the cells aggregate together based on not being able to attach to those plastic surfaces. Hanging drop involves suspending cells in a small droplet of media, and those cells then aggregate by gravity. And then, of course, for our dynamic systems, probably the most commonly used being orbital shakers, where flasks or plates are placed on a planar shaker, and the fluid motion of gentle swirling keeps those 3D structures in suspension as they're being cultured. And then finally, our bioreactor class, which are typically impeller or stir-driven culture systems. Generally, these are used for more large-scale production or larger organoid systems. And ultimately, these methods are used to create organoids for a variety of applications. So today we're talking about disease modeling and drug and toxicity assessment. But as I mentioned, these organoid models are useful in personalized medicine and regenerative medicine applications as well. And in order for these models to be adopted for these purposes, we first and foremost need them to be phenotypic and functionally relevant to the tissues they're modeling. And this is where a lot of the efforts in the last decade have been to establish methods and protocols that generate tissue-specific organoids. But as we start to consider moving these models into regulatory environments, we also need to center those protocols and methods around technologies that enable resource efficiency and scalable culture systems, and very importantly, are reproducible, both across different cell lines, but also from user to user. And this is where the real challenge of organoid culture lies. While each of these methods can be used to generate spheroids and organoids, each of them also come with inherent disadvantages that have to be considered before they can enter into regulated environments. A lot of these methods involve the potential for variability, including user-dependent variability, and some of them are very low efficiency, which are all reasons why regulatory acceptance can be in question. Today, I'm going to introduce you to a proven technology. The Zero 3D incubator and bioreactor has been widely adopted in European markets and is now available in the US from Cell Microsystems. The Zero 3D is a high-yield benchtop bioreactor that enables consistent cultivation of suspension-grown spheroids and organoids and combines automation with ease of use and culture conditions that can support the biological complexity of organoid culture systems. Scientists are using the Zero to generate a very diverse landscape of cell types, and each of these images represents methods that have been developed using the Zero. So this highlights the broad applicability of this technology for 3D culture. For the purposes of today's webinar, since we're talking about drug discovery, I'm going to be focusing on 3 critical models for the drug discovery process: the brain, heart, and liver. These models are critically important because the tissues are difficult to source. They're common sites of toxicity and adverse events, and ultimately, each of these tissues are required for safety testing. So for our first model system, we're going to be discussing cerebral organoids. These organoids can be used for a variety of different diseases. Today, I'm going to be talking about them in context of cancer. The median survival rate for cancer, brain cancers, are around 10 months. And in addition to that, only 3 to 7% of drugs that are developed to target cancers are actually successful. This is partly due to the fact that recapitulating the tumor microenvironment in vitro is very complex and challenging to do. In addition to that, neurotoxicity is a big concern of many drugs. In this first case study, I'm highlighting the work of Wolfram et al. This research group sought out to establish this end-to-end pipeline of 3R compliant mouse model systems for brain cancer modeling. Now, I know I just said the current push is for human-based models, but these fit-for-purpose animal models still play a very significant role in the drug discovery process because they can bridge the gap between human in vitro data and animal testing, and they can also feed back into those in silico modeling systems using historical toxicity data. So I really enjoyed this paper because it built this beautiful pipeline of in vitro models that are increasing in complexity, starting with 2D cancer cells and differentiated neurocells, and then all the way through building out these advanced core models. What I'll be highlighting today are the model systems they used the Zero 3D for, and that was for the cultivation of adult neural stem cells as neurospheres, differentiation of those neurospheres into cerebral organoids, and then the long-term culture of those organoids. For neurosphere production, they compared the Zero 3D method with their traditional ultra-low attachment plate culture method. They found that they could culture up to 50 million adult neural stem cells in one Zero tube using just a very small amount of media. And this was more than 20 times the capacity of what they could do in their ultra-low attachment plates. In addition to that, they found that by culturing in the Zero, they had reduced clumping of those neurospheres, and this meant that they could less frequently passage the neurospheres compared to their ultra-low attachment culture. Ultimately, the protocols that they developed enabled a very efficient system to scale up neural stem cell production And they could use those neural stem cells for either 2D high throughput assays or for organoid development. So I mentioned they developed protocols to differentiate those neurospheres into mature cerebral organoids. And here in this image, you're looking at the neurospheres as they differentiate into organoids. And to begin to characterize those organoids, they did sectioning and staining of the organoids. And you can see here that they exhibit the canonical neural maturation markers for astrocytes, neurons, and oligodendrocyte cells. As they continued to culture those organoids long-term, they had checkpoints where they continued to monitor their differentiation and maturation. And here you can see the 8-week organoids are positive for both neural differentiation markers GFAP and NUIN, as well as neural stem cell markers Fomentin and SOX2. Now, after developing those protocols, when they compared the differentiation across the two methods, being either the sero or the orbital shaker, what they found is that in the sero, they could culture the organoids for much longer, more than 5 months. In addition to that, the sero-grown organoids had structural characteristics that more closely resembled the in vivo tissue. So they were, number one, more spherical in shape, which you can see here, the difference between the sero and the orbital shaker. In the sero organoids, the distribution of Ki67, indicating proliferation, was found throughout the organoid body, whereas in the orbital shaker organoids, that Ki67 was only present on the rim of the organoid. They also found limited apoptosis indicated by cleaved caspase 3 in the sero-grown organoids. So after developing their organoid models and characterizing them, they wanted to employ them in their more complex brain cancer modeling. To do this, they generated assembloids using the mature cerebral organoids and a GL261 glioma cell line, which they made spheroids from. When they typically, when they culture these assembloids in an orbital shaker, they expect less than 10% of the surface area to be interconnected between that cerebral organoid and the glioma spheroid. However, when they cultured them in parallel with the sero 3D, they found that the interconnected surface area increased by more than 50%. This increase in interconnectedness better reflects that tumor tissue interaction. And ultimately, as they continued to study these, they found that they could more accurately model that tumor microenvironment when they created these assembloids in 3D, in the sero 3D. So altogether, this publication demonstrated a very easy to use method using the sero to generate hundreds of organoids per tube using less media compared to the 6-well plates. The organoids that they generated had a more reproducible morphology, and they maintained those proliferative cores. And ultimately, the sero also enabled them to culture their organoids for longer. The sero-grown organoids were cultured for more than 5 months, whereas they started noticing apoptotic cores in the orbital shaker cultures around 3 months. Altogether, they found they could culture 1.6 times longer using the sero 3D, that it supported 15 times more organoids per unit, and required less hands-on work by their scientists compared to 6-well suspension plate methods. Our next model system that we're going to discuss are cardiac organoids. iPSC-derived cardiac models are invaluable in studying disease and drug toxicity testing. And that's because more than 13% of withdrawals from the drug discovery process are related to cardiotoxicity. And it's well known that 2D and static 3D cardiomyocyte production is an inefficient process. I've seen it in my own hands. I've heard it from researchers in the field over and over again. There's often operator and cell line dependent inconsistencies where you have your 2D cardiomyocytes beating in a lawn of partially differentiated or undifferentiated cells. So suspension culture for this differentiation method not only offers scalability, but many times we see that suspension differentiation actually enhances the differentiation process. And that's what the researchers in this next study sought out to investigate. So importantly, they used 2 iPSC lines for this study. The first was an established cell line from a European cell bank. The second was a reprogrammed cell line from their own lab. And this is really important because, as we mentioned, we need to ensure that the methods that we're creating to produce these organoids are reproducible even across different cell lines. To compare the methods, they generated these cardiospheres using their standard 96 well plate hanging drop method, and then, of course, the Zero 3D, which created that dynamic suspension environment. In addition to generating the cardiospheres, they also wanted to test dissociation methods because, as I mentioned, many times these 3D differentiation methods are much more efficient than 2D. And so they wanted to see if they could dissociate those cardiomyocytes and use them in 2D high throughput screening methods. So here in these images, you can see on the top is their reprogrammed cell line, and on the bottom is that iPSC line that they sourced. And you can see that for both lines, the cardiospheres demonstrate similar size characteristics. And in the bar graph here, you can see that they also have a similar growth rate with the Zero method. And importantly, I'd like to point out that the error bars for the diameters here are very small. And that's important because that demonstrates that the suspension environment of the Zero 3D supports homogenization throughout the culture. To begin to characterize the cardiospheres, they did qPCR. So in gray, you're looking at the hanging drop method, and red is the Zero. And you can see here they had a similar reduction in the pluripotency markers over the first couple of days. However, with the Zero, they noticed that they had faster mesoderm specification. So this peak here and here coming on sooner in the Zero 3D compared to the hanging drop. And in addition to that, they also noticed that their markers for cardiac specification and mature cardiomyocytes were elevated in the Zero 3D compared to hanging drop. Ultimately, this indicated that that dynamic culture environment better supported the cardiomyocyte induction. And you can see here in these videos that for each of the cell lines that they were testing, they get this canonical beating morphology with their Zero 3D protocol. So to further characterize these cardiospheres, they looked at protein expression by staining the cardiospheres. So here in this right panel, you're looking at the Zero 3D, and you can see that for the structural proteins they were interested in, actin and desmin, that the Zero 3D cardiospheres have elevated expression, which is evidenced here by the bar graph. Further, they looked at connexin 43 expression. Now, connexin 43 is involved in the propagation of the action potentials, ensuring that synchronized contraction of the cardiospheres. And they found that in the Zero 3D, the expression of connexin 43 was more than 10-fold that of the hanging drop method. So altogether, again, pointing that this dynamic culture environment induced a higher level of physiological maturation. And the researchers actually attributed this to the better distribution of oxygen and nutrients generated by that medium flow in the Zero 3D, improving their cardiac differentiation. So I've shown you previously that they found that the Zero produced more mature cardiomyocytes compared to the static culture. And they also showed that 3D propagation of cardiomyocytes was more efficient than 2D. But it's well known that 3D things are more challenging to dissociate. So they wanted to test different dissociation methods to see if those mature cardiomyocytes could then be replated and used for their 2D high throughput screening. So you can see here, they were successfully able to dissociate those cardiospheres, and when they replated them, they maintained their functional phenotype after that replating. Altogether, this study showed that the researchers were able to significantly improve their differentiation and maturation of those cardiomyocytes compared to their hanging drop method. Not only that, but they were able to create this workflow that efficiently generated a very high number of viable functional cardiomyocytes. And in fact, they found that in just one serotube, they could generate this to generate the same number of cardiomyocytes as that one tube. They would need more than 10 T75 flasks to do it 2D, or 196 well hanging drop plates. So a great improvement in the efficiency of generating those cardiomyocytes. So our final model system is maybe the most critical, and one that is certainly near and dear to me personally, is the liver organoids. Drug-induced liver injury is known to be a very major cause of drug failure that spans the entire process. And markedly, 30 to 50% of drugs are recalled after they are approved by the FDA due to drug-induced liver injury issues. So currently, when testing, 2D primary hepatocytes are the gold standard for toxicity testing. We know that 2D cultures do not represent the important spatial arrangement, but in addition to that, with hepatocytes, they have very limited viability in culture, and in fact, they lose their metabolic competency within 24 hours. So not only are the exposures very short, but oftentimes when you're getting these from donors, they don't represent the genetic diversity. You're waiting on a single donor to do your testing. And overall, the goal is to generate 3D models that offer improved viability of those cardiomyocytes, but also better fidelity for the in vivo exposures, having the ability to culture them for longer and also get better diffusion. And in addition to developing the 3D models, there's a push for creating biobanks of ready-to-use model systems. And this is important because you can have available to use these pre-qualified and tested biobanks that can be available as needed, rather than having to wait for a donor to come along to do your toxicity testing. In this study, the researchers used a gene-edited CYP3A4 nanoluciferase iPSC line. This reporter cell line was used to generate hepatic organoids in the sero. And as I mentioned, they were incorporating this idea of generating a biobank of ready-to-use organoids. So they tested cryopreservation methods of progenitor organoids at day 8, as well as mature organoids at day 21, and throughout the process did a lot of batch QC analysis. Now, as I mentioned, they used the sero to not only generate these organoids, but also directly reseeded them into serotubes for recovery from frozen. So here in the images, on the left, you have the day 8 organoids. On the right, you have day 21. And you can see them before and after freezing and out to 7 days in culture. And with both of them, there's an increase in organoid diameter, and they seem to recover well from thaw. Using that nanoluciferase reporter, they looked at CYP3A4 expression, and for both organoid types, the day 21 down here on the bottom and day 8, they had a similar expression of CYP3A4 immediately after recovery. However, the day 8 organoids showed a more similar and consistent profile of CYP3A4 expression as they were further differentiated compared to the day 21. So they decided to further characterize the progenitor organoids for their competency. not just showing that they could be frozen and thawed, but they did qPCR analysis to look at canonical hepatocyte markers. And here you're looking at the qPCR, a few of the qPCR targets normalized to primary hepatocytes. And as you can see, in gray being the control and blue being the thawed, they had similar expression profiles of the progenitor markers as well as mature markers. So decreasing in those progenitor markers over time, whereas the mature markers increased over time, similarly between the control and thawed organoids. So this further supported that those organoids could be recovered, and they continued to differentiate them into mature hepatocyte organoids. And here you're looking at immunocytochemistry staining of those organoids 14 days after thawing. And other than HNF4 alpha here, you can see both for the non-frozen controls and the organoids that were cryopreserved, there are similar staining profiles between the thawed and non-frozen controls for these mature hepatocyte markers, indicating that the cryopreservation did not impact the organoid's ability to continue to differentiate. So altogether, this group developed a very efficient protocol for scalable differentiation of hepatic organoids. They were able to achieve 5,000 to 7,000 organoids per tube, and the tube was not even at capacity with just 20 mils of culture media. They determined that the progenitor organoids could be cryopreserved and then resumed into culture and continued to differentiate out to 21. And this offered that faster path for organoid drug screening, eliminating the need for waiting on donor testing. And ultimately, they found that with the sero protocols, they could create this biobank of organoids for drug screening. So early in the presentation, I outlined for you some of the required elements for generating these reliable organoid models. And using these case studies, I hope I've convinced you that the Sero3D is a superior platform for developing more mature and functionally relevant organoids, to develop methods that are both efficient and scalable, as well as a culture environment that supports reproducible and consistent organoid culture. And these case studies were just a very small subset of the broad landscape of publications that are available. So by now, you're probably wondering exactly how the Sero3D works. It is a benchtop incubator and bioreactor, and it has a very small, convenient footprint. Many of our customers have multiple instruments, and they can very easily sit front to back on a standard benchtop and only take about 1.5 to 2 feet of bench space. Within one instrument, there are 4 independent tubes that can be cultured. This allows you to optimize and to set different rotational parameters and timing of those parameters for the optimal growth of your organoids. So essentially, you can run 4 independent experiments congruently. The chamber is HEPA filtered for sterility, and the controls offer logging of temperature and CO2, which is convenient and impactful for GMP environments. In addition to those logging methods for temperature and CO2, there's also the opportunity to monitor pH, and this can be done online. And many of our customers find this useful when they're generating SOPs so that they can make consistent decisions about their cultures, like when to change media or about the viability of their cultures. But the real magic behind that gentle suspension environment that I've been telling you about is the sero tubes themselves. The sero tubes look and feel like a 50 ml conical tube. They have a filtered cap for sterility assurance. And even though they look like a 50 ml conical tube, they allow for a flexible culture volume between 7 and 50 mils. And that flexible volume allows users to not only scale their organoid culture methods, but also supports media efficiency. So if you don't need thousands upon thousands of organoids, or if you're designing your SOPs, you can do so using much less media, which is not something that other bioreactors allow you to do. Within each tube are fixed homogenization baffles. These baffles offer that gentle levitation without the need for any impellers or stirs. And ultimately what that means is that your organoids are cultured with very low shear forces. In addition to that, the media is consistently moving throughout the tube, offering that optimal nutrient supply and media movement, which enhances your organoid maturation. And as I mentioned, these are much like a 50 ml conical tube. They're carried in and out of a standard biosafety cabinet, so you can very easily change media, but also sample from the tube at various time intervals for your organoid QC checkpoints. So to do those media changes, it's really quite simple. You stop the rotation of the tube. The tube is allowed to sit in that controlled incubator environment. And importantly, you only have to stop the rotation of the tube that you're interested in handling. So unlike an orbital shaker, each of these tubes can be started and stopped independently. So after the organoids have settled to the bottom of the tube, you aspirate the media, add fresh media, and return it to culture. And ultimately, I found and our customers find that we're typically spending less than 5 minutes once or twice a week to handle thousands of organoids using these methods. So here you can see the instrument live and in action. One thing that I really enjoy about the Zero is that it has self-contained controls. So you can see the touch screen here, where you can very easily access and control each individual tube. As I mentioned, each of the positions is independent of each other. So in this video, you can see these tubes are rotating at different timing and speeds. And that's important because it allows you to culture different organoid types or to build these pipelines where you have organoids culturing at different intervals so that you always have organoids ready to use. In addition to the Zero, here at Cell Microsystems, we have another technology in our portfolio called the Cell Raft technology. And combining these two technologies, it's enabling users to generate custom 3D cell-based assays at scale. So we're getting that high yield cultivation using the Zero, and then the Cell Raft technology offers the opportunity for image-based software-guided selection and isolation of individual spheroids or organoids into 96 well plates for downstream testing. And both of these instruments are available for purchase through Cell Microsystems. Now, in addition to having the instruments available, we also do lab services. This can look like custom cell line development, whether that's 2D cell lines or organoid development. Our services allow you to accelerate your data generation by utilizing our trained scientists and ultimately improve your team's productivity. And then what we tell people is when you're ready to buy an instrument, it's a very easy gateway to ownership. We can help you develop the methods here with our services, or if you buy an instrument, and then assist you as you start to optimize your protocols for your 3D cultures. If you're interested in more information about either of the technologies, I'll invite you to our website. And with that, I will conclude and turn the floor back over to Lisa, who is going to field the questions from our audience.
Thank you so much, Dr. Stern, for that informative presentation. Before we get started with the Q&A session, I do have a quick poll for you that I'm going to launch on your screen now, which we would greatly appreciate if you would take a moment to answer and to allow some of the questions to come in. We've already had a bunch of questions come in, but as a reminder, please feel free to ask your question in the chat box, and we will get to as many questions as we can. And if you provide your information and we don't get to your question, we will happily send you an email to follow up. So let's get started with Dr. Stern. So the first question is, I've never grown my organoids in suspension. How do I know they'll grow in the sero?
Yeah, that's a justifiable question. So what I can tell you is that we've worked with a lot of scientists to take protocols that have never been grown in suspension and transfer them to the sero. This can look like some optimization of those rotational parameters, as well as certain organoid models rely more heavily on the extracellular matrix to form their 3D structure. But the great thing is that whenever you purchase an instrument, you also have access to our scientists and our expertise in using the instrument and adapting these workflows. So one of the things that we enjoy the most is helping our customers transfer their protocols, develop new protocols. And honestly, we've even worked with some researchers who are growing organoids successfully in the sero that they've never been able to grow in standard culture methods. So while it can seem a little bit of a hurdle to kind of do that optimization, we're here to help you with it, and we feel very confident in our ability to transfer those protocols to the sero and suspension.
Excellent. So in that vein, was there an extracellular matrix used to create the organoids? And if so, how is it added into the sero 3D?
Yeah, also a great question. So it really depends on the organoid model. And I know that's kind of a tough way to answer that, but it's still biology, and there are certain organoid types that require more structural support for their 3D formation than others. So to answer the question in kind of a broad way, we have worked with scientists to create methods using what I call like a dilute suspension approach. And essentially what that means is that we are spiking in matrigel or another extracellular matrix or hydrogel into the media as we start the tube with those organoid forming cells at a very low concentration. So this can look like 2 to 4% of what you might normally use to culture your organoids. So a considerable savings if you're trying to grow your organoids in less ECM. On the other hand, there are some organoid models, like I said, that do require that more rigid structural support to form their 3D structures. And so we have some customers that culture their organoids essentially in like floating domes. And while that might seem a little bit counterintuitive, what they find is that even though they're still embedded in that extracellular matrix dome or kind of bubble, so to speak, that the dynamic environment of the sero still enhances the maturity of their organoids and the culture process of their organoids. So whether they're using less media and growing more organoids, or they're getting more mature organoids, they still find the benefit of doing that suspension culture. So it really depends on the model, but like I said, that's something that our scientists and our team is very able to help you with as you kind of overcome that initial culture hurdle.
Great, thanks, Alyssa. So the next question is about the instrument itself. Being a benchtop incubator, is it tri-gas capable? So if you want to grow cells in a hypoxic environment to better mimic the tumor environment for cancer studies, is that possible?
Yeah, great question. We get that one a lot. So it is a single gas input, but I know that there are some users that are interested in doing those hypoxia studies. There are some users that do it based on media methods. And then there are also options to have like external gas mixers, where essentially you have a gas mixer that is incorporating the tri-gas and then feeding it into the sero, just with one line. But the instrument itself only has one gas port.
Got it. And in that same vein, how do you measure pH in the sero 3D?
Yeah, so within the incubator environment, there is essentially optical sensors that measure the absorbance of the media. So to measure pH, it would require a phenol red containing media. And essentially what the system does is measure the absorbance of that media and then can translate that with some input from the user about their specific media to a pH value, or just give you that absorbance readout so that you can loosely translate it to pH. So there's two different methods of measuring pH. One is a little more specific, which includes that calculation of the pH based on absorbance, and then the other is just absorbance. But that's all done by the optical sensor.
Excellent, thank you. I love this question. Can this bioreactor be used for co-culturing different cell types?
Yeah, of course. The one example we showed today was, you would call a co-culture in that assembly model. So the assembloids there were formed in plates and then transferred into the sero for long-term culture. But absolutely, you can co-culture different cell types or organoids and, like an immune cell type, whatever it may be.
Great. This one is about our custom organoid services. Can you make organoids from patient-derived iPSCs that we provide you?
Yes. Most of our customers do tend to have a specific cell line that they're interested in us using, and we can definitely do that. Like I said, this is all of our organoid services are bespoke or kind of tailored to what our customers need. So we'd absolutely be interested in discussing those services if you have something you're interested in.
Awesome. This one wants to know if you have how big this one says, how big are these spheroids and organoids in terms of millimeter sizes? How can you maintain the uniformity between them?
Great question. So the size likely depends on both the proliferative capacity of your organoids. Many a times as they are differentiating, their proliferation might slow down, but also kind of the input. And so getting at that, making homogeneous cultures. Again, not to point the finger at biology here, but there are some organoid types and there are some workflows where we can just start the cells as a single cell suspension and the sero and get a nice homogeneous culture. And there are other differentiation protocols or organoid protocols that benefit from pre-forming those aggregates or those early organoid stages and then transferring them into the sero after they've been pre-formed so that you can control the size more to your specific ability. Because of course the aggregation and suspension in the sero is in suspension and it is somewhat random. Now we can adjust rotational programs and cellular input to try to achieve specific sizes of organoids early on. But as far as, you want to ensure that you have a very homogeneous culture, there's also early plate stage methods that we can do and then transfer into the sero for that long-term culture.
Excellent. Let's see, the questions are coming in fast and furious. Can you please explain the dissociation process and any effects on viability, presumably to get the cells into the serotubes?
Sure, yeah. There are several different methods of dissociation. The one example that we talked about today used trypsin in a few different enzymes. I would say it kind of depends on the organoid type that you're working with, what method or what solution we would recommend to dissociate those based on what is optimal for that particular organoid type. But essentially, what you can do is either if you're harvesting, you can let them settle by gravity, or if you're literally just doing some sort of single cell dissociation for downstream use, you can make a pellet of those organoids and then dissociate them, wash and dissociate them after the culture has stopped. There is also a way to do the dissociation in the sero by rotational parameter programs that will kind of keep that organoid solution spinning to help sort of mix that solution as the organoids are dissociating. Again, it kind of depends on what the end goal is and what organoid type you're working with.
Great. I think we maybe have time for one or two more questions. What is the general max throughput of organoids per tube? How long have you tested the culture of organoids in the sero for up to days or weeks?
Okay, so the first part of that question on the organoid number per tube. Again, biology, a little bit of that depends. Maybe how long you're culturing them, how large they are, and sometimes can even be linked to their metabolic capacity and how quickly they're utilizing that media. I would say on the order of magnitude of hundreds to thousands per tube. And again, remember there's that flexible volume of media. So we can really tailor the workflow to adjust to the scale that you're interested in. So we have some customers that say, you know, I only need 100 or 200 organoids. Great, that works perfect with these smaller volumes, and we can adjust your workflow to conserve media while still using the sero. And then other customers of ours who are doing like organoid biobanking are really trying to get the max capacity from the sero in which they can up that volume to the 50 mils. And they're culturing 40, 50,000 organoids in a single tube to then create those biobanks of organoids to cryopreserve. So it's very flexible on the organoid output and depends a little bit on what your application or workflow is. The second part of that question was, sorry, Lisa, can you remind me again? The second part was on the
how long can they be maintained?
Yeah, of course. Weeks, days to weeks to months is the short answer. So there are some organoid models that are highly proliferative and need that rapid passaging. And so some organoid models, like the adult stem cell derived organoids that need passaging every 7 to 10 days, that's how long you would culture them before passaging. Other organoid models, like we've done cerebral organoids and cultured them for months in the sero. It really depends on your model specifically and what you're interested in doing with them.
Okay, great. And I think we have time for one more question. This one says, is this just for generating the organoids, or can you also test compounds in the sero?
There's no reason you can't test compounds in the sero. So I would say it's flexible depending on what your application and your downstream endpoint is. So, you know, again, with that media volume, you have the ability to culture hundreds to thousands of organoids. If you're interested in looking at population level responses to, you know, a drug or a toxicant or something, you can absolutely do that in that, you know, population level way.
Excellent. Okay, well, Alyssa, thank you so much for answering all of those questions. And honestly, thank you all for joining us today and for the really active discussion. There were so many questions. And we want to thank Alyssa for sharing her expertise with us today. Please remember, as the webinar closes, we'd love for you to answer our short 3-question survey when you close out of the webinar. If you would like to learn more about any of the tools and workflows that Alyssa discussed today, please feel free to reach out to us via email or via our website at cellmicrosystems.com. And a recording of this webinar will be sent to all registrants, so please keep an eye on your inbox. And as a reminder, keep asking questions. If there's anything that we didn't get to that you'd like an answer to, please provide your contact information, and Alyssa will get back in touch with you to answer everybody's questions to make sure everyone gets a chance to be heard. We want to thank you again for your time and for your attention, and have a great day.